Okay, everybody, I believe the time is up. Right now is 9 past 10 local time in Indonesia. Good morning, distinguished guests, respected speakers, college researchers, and all participants joining us today from Indonesia, South Korea, and beyond. It is my great pleasure and honor to welcome you to the Deep Sea Science Forum toward Deep Sea Mission 2045. On behalf of the Research Center for Deep Sea National Research and Innovation Agency, or BRIN Indonesia, I would like to warmly welcome all of you to this forum. In particular, we are pleased to have Professor Myung-hee Kang from Gyeongsang National University joining us today. I'm really sorry if I have misspelled that because uh, sometimes my tongue is not very good. Professor Kang Myung-hee, Wang Yong Hamnida, and welcome to Indonesia to the Deep Sea Science Forum. The deep sea represents one of the largest and least explored environments of our planet. It plays a critical role in regulating our ocean systems, supporting marine ecosystem, transporting energy and materials, influencing the global climate system. At the same time, the deep ocean remains a challenging environment to observe and understand because of the fast scale and extreme condition and complex interaction between physical, chemical, geological, and biological process, especially in biological with the time today. For this reason, advancing deep sea science requires not only sophisticated technologies and high quality observation, but also strong collaboration across scientific disciplines across national boundaries. Acoustic observation, oceanographic measurements, biological studies, environmental monitoring, and geophysical surveys can provide different pieces of the same complex picture of the ocean. By bringing this approach together, we can develop a more comprehensive understanding of the deep sea environment and its response to rapidly changing ocean. Today's forum provides an excellent opportunity for us to exchange knowledge, share research experience, and explore new possibilities for scientific collaboration. We have two distinguished researchers who will present their work on very interesting and complementary topics, from the integration of acoustic, biological, and environmental factors in understanding tropical tuna to the transport and accumulation of microplastic from the surface to the deep sea environment. I believe that these perspectives are highly relevant to our shared efforts toward a more comprehensive understanding of the ocean and to the broader vision of Deep Sea Mission 2045. My name is Omar Mufti from the Research Center from Deep Sea BRIN, and it's my pleasure to serve as your moderator for today's session. I hope we can have an engaging and fruitful discussion, perhaps more importantly, and identify opportunities for future collaboration between researchers and institutions. Once again, welcome to the Deep Sea Science Forum. Thank you for taking the time to join us today. And without further ado, let's begin our program. To officially open today's forum, it's my great pleasure to invite Professor Aan Johan Wahyudi, Head of the Research Center for Deep Sea BRIN, to deliver the opening remarks. Professor Aan, the floor is yours.
Thank you very much, Pak Omar. And good morning, distinguished speaker, colleagues, researchers, students, and participants joining us today. On behalf of the Research Center for Deep Sea National Research and Innovation Agency of Indonesia, it is my pleasure to welcome you to the Deep Sea Science Forum as part of our continuing journey toward the Deep Sea Mission 2045. Today, we bring together two topics that may initially appear quite different, yet the topics are connected by the same fundamental principle, which is the ocean is an interconnected system. As we know that ocean current transport heat, nutrient, organism, and biological productivity across large distance, and this process shape marine habitat and influence the movement and distribution of highly mobile species such as tuna. At the same time, the same circulation can transport human-generated materials, including pollutant and plastic, connecting activities at the surface and along our coast with environment far offshore and ultimately or even the deep sea floor. This is why understanding the ocean requires an increasingly integrated scientific approach. And our first speaker, as mentioned by our moderator, this is Professor Myung Hee Kang from Gyeongsang National University, South Korea, will present an integrated analysis combining acoustic and environmental information to better understand tropical tuna in Western Indian Ocean. This is an excellent example of how different observations can be brought together to understand the relationship between marine organism and their dynamic environment. And the second speaker, we pleasure to have Dr. Koryati Manulang from the Research Center for Deep Sea. She will take us along another pathway from surface waters through Indonesian through flow toward the deep ocean to examine how microplastic are transported, taken up by organisms, and eventually accumulated in the deep sea environment. These two presentations will illustrate important message for deep sea science. What happened at the surface does not necessarily remain at the surface, and what happened in one part of the ocean may have consequences far beyond that region. This perspective is also central to our deep sea mission to 2045, which is exploring the deep sea should not simply mean going deeper. It's mean understanding the connection between the atmosphere, surface ocean, water column, marine organism, and seafloor, and connecting observation, technologies, data, scientific discipline to build a more complete understanding of our ocean. I therefore hope that today's forum will not only provide new scientific insight, but also stimulate discussion and opportunities for future collaboration, particularly among Indonesian and international scientists and the next generation of ocean researchers. And finally, I would like to express my sincere appreciation to both of the speakers today and also the moderator that work together with us. And also, I would like to thank everyone who joined us in this forum. Welcome to the Deep Sea Science Forum. I wish you all productive and inspiring discussion. Thank you very much.
Thank you very much, Professor An, for the opening remarks and for setting the stage for today's forum. Ladies and gentlemen, we will now move to introduce our presenter. Okay, it's my great pleasure to introduce both of our speaker. The first is Professor Myung Hee Kang from Gyeongsang National University of South Korea. Professor Kang currently serves as professor at Gyeongsang National University. She holds a PhD in visual science from the Tokyo University of Research, and she brings extensive expertise in acoustic data analysis and fishery acoustics. She previously served as technical manager at Miriax Limited in Australia, where she provided technical support and consulting for acoustic surveys and biomass estimation projects around the world. Okay, she is also invited member of the working group on fisheries acoustics, science and technology. Today, Professor Kang will share her research entitled First Integrated Analysis of Acoustic, Biological, and Environmental Factors Affecting Tropical Tuna in the Western Indian Ocean. I believe this topic provides an interesting perspective on how acoustic, biological, and environmental observation can be integrated to better understand marine ecosystem and tropical tuna. After all, we all love fishing. And for the second speaker, uh, it's my pleasure to introduce you to Ms. Dr. Koryati Manulang from the Research Center for Deep Sea National Research and Innovation Agency, BRIN, Indonesia. Dr. Manulang is an associate researcher at the Research Center for Deep Sea BRIN. She earned her PhD in marine pollution from University of Indonesia in 2024. Prior to that, she completed the double degree master's program in marine science at University of Ponegoro and University de La Rochelle in France. Her research focused on primarily on marine pollution, with particular emphasis on the concentration, distribution, transport, and environmental implication of microplastics. Her field research covers several important Indonesian waters, including Indonesian through flow, the Bangai Sea, and small island in Maluku. Today, Dr. Manlang will present her research entitled Microplastics from Surface to Seafloor in the Indonesian Through Flow, Oceanographic Transport, Biological Uptake, and Deep Sea Accumulation. This is a highly relevant topic today, particularly given the importance of the Indonesian Through Flow and its role in transporting materials through Indonesian seas and into deeper ocean environment. Okay, Professor Kang, first the first presentation. Thank you for very much joining us today. Can you share the screen, or we can share your presentation also? It's
Can you see my screen?
yes, of course. I can your screen all right. Perfect, perfect, perfect. The floor is yours.
Hello, I am Yonghee Kang from Gyeongsang National University in South Korea. And this is, my talk, acoustic, and I'm eco-sounder in person. And this time in biological is zooplankton sampling and environment in vectors. So, we integrated, those three elements, for understanding tropical tuna in West Indian Ocean. And here is Indian Ocean. And the IOD is the Indian Ocean dipole, and which is similar to El Nino and La Nina. So it affects climate and weather and ecosystem, and as well as people's inner life. So IOD is important in a matter to understand IOD. And so people, normally oceanographers, and they studied two different reasons. One is on west side, the other is on east side. I guess this is more like your area. I believe in many Indonesian scientists involved in East IOD zone. So this time, we focus on west side. And here is in the west side of the Indian Ocean, and specifically two countries. The Seychelles is the name of the country, and Togo is another country. And our study area is SCTR, and which is Seychelles, Togo's thermocline ridge. This is an area, and latitude is in a 5 and 12s. It's not exactly that reason, but normally people assume that is 5, 12, and then longitude 55, 19. This is upwelling, thermocline upwelling region. So it is relatively cool and then very rich and productive area. Just let me check whether I can do I can, the mouse, So like Indian Ocean is, way in from, South Korea. And, why people, some people in the mind, think, why we come down here to study. And here is one example. And Jamstek is a Japanese scientist study about Indian monsoon is coming through East Asia up to Korea and Japan. So that Indian Ocean is affecting Korean, weather. So I mentioned about acoustic, and I am acoustic person. And here is sound scattering layer, SSL. We call it SSL. And typical research in a vessel has sensors. This is one example in 38 kilohertz, and send sound and receive an echo and detect like this scattering layer. And sometimes we call shallow scattering layer, deep scattering layer, but overall sound scattering layer. It has various species, and micronecton, and there are various species, and also zooplankton. So SSL has a micronecton as well as zooplankton. And this Indian Ocean study, we joined 2020, but we used the 2019 data, and it was the first publication. So we would like to broadly understand what's going on in SSTR area. So we used the two frequency and 18 and 38 kilohertz. So we saw up to 1,000 meter and target within in SSTR area and beyond in SSTR area. And here is some people published already, and they use 18 and 38 kilohertz and categorized, acoustically categorized. And so we used those in the tables to understand, and some, like for example, and small non-swim bladder fish or large non-swim bladder fish or swim bladder fish, etc. And the 2019, it was a cruise track, and one in 60E, and right side is a 65E. And this is a 8, 5 about 5, 12 in S. So we use this is STR area, this is non-STR area. And here is in echogram, and we call DVM, diurnal migration, the movement and vertical movement. So here is in 60E, and 60E is like this, and 67, sorry, 67 is right there. And you can see in one day typical DVM pattern. And we used the 2021 data, and this time up to 150 meter, and used 38 and 120 kilohertz to focus on and fish and microplankton. So it was a COVID period, so we didn't have a proper cruise track. So we just showed briefly in horizontal distribution of left is a microplankton, right is a fish. And using 23 data, and SSL is a layer. It's very hard to know the matrix. So we would like to, we select SSL core area to understand morphological features and distributional dynamics. So here is example matrix of SSL, length, thickness, center of mass and depths, and how the altitude from the sea bottom. And Indian Ocean is well known about tuna. So here is example yellowfin tuna. And the Indian Ocean is the second largest tuna fishing region worldwide. So 2023, and they had 1.2 billion tons of tuna catch, so which is 6.5 million US dollars. And IOTC in Indian Ocean Tuna Commission classifies skipjack and big eye yellowfin tuna as a tropical tuna species. Here are some pictures: big eye, yellow, and skipjack. So objective of this study, and we would like to see the relationship between tuna catch and prey and biomass. And so far, there is not that research. And we would like to integrate IOTC tuna catch data and ecosounder data, and then environment data. And we done zooplankton sampling so that we would like support the idea, the relationship between tuna and the prey organisms. So yeah, basically, want to understand more about trophic dynamics in West Indian Ocean. And here are study area, and all the circle is a tuna catch in 2023, and the cross specifically in May and June, which is the acoustic survey period, and the circle, the green circle is a cruise track, So we can see cross-track here, and then IOTC tuna, the catch, and the 3 points is a multi-net sampling station, 19S, 8S, 19S, 8S, 2S, in which are the multi-net sample. And we use the RB Isabo is the name of a vessel, and acoustic system is EK80. And we used only one frequency, like representative in frequency, in 38, calibrated 38 kilohertz. And this is a multi-net sampling gear. And I mentioned about 3 locations in the same latitude, but 3 different latitude, but same longitude. And IOTC data, and it has many different data, so you can download. So we typically collected in the data and big eye, yellowfin, and skipjack, and the area we can set, and then time is 2023. And this data has only length information, and length in a month, and fishing method, location, but it doesn't produce in a catch. So we use the equation and body length and body weight to convert tuna catch. Here is equation, tuna L length and weight equation from FAO, and species, gear type, and equation. So that once we know in L from IOTC data, so we can convert their weight. So we can add up and total catch. The marine environment properties, we had a Copernicus Marine Service, and we select the time, which is the same as the acoustic survey, and we collect temperature, salinity, chlorophyll, and DO. And we can retrieve in that CDF format, and it has a 28 depth layers, but we select in 2. One is a 47.4 meter, and which is above thermocline, and one is below thermocline in 130.7 meter. And data, especially acoustic data, we use the EcoView, and clean data using various noise removal algorithms. At the end, in acoustic data, and we use the mean volume backscatter strength, which is MVBS, and it is a key acoustic metric to quantify the density of the underwater targets. And we also use ArcMap to visualize, and specifically we use the buffering and within a 30-degree radius. And here is the circle I mentioned about 30-degree. 30-degree radius based on the tuna catch, so that we can know tuna catch and that environment data inside this circle, as well as those are MVBS. And I mentioned about acoustic backscatter, and which we use the apply in organisms in biomass, and those are example. We used some other bit to visualize and then to connect the 3 elements, 3 data. And we use the GAM, the generalized additive model, to see nonlinear relationship between tuna catch and prey abundance, and then tuna catch and environment in the variables. And this is I mentioned about again, and the GAM, and the reason is to see tuna catch and MVBS, and tuna catch and environment at 47.4 meter, which is above thermocline, and tuna catch and environment elements at 130.7 meter, which is below thermocline. Here are result. And this is in a month, and then converted in a weight. So we can see we done survey May and June is a quite low the catch. And this is the monthly variation, body weight and fork length, and blue pin. So big eye, sorry, not blue pin, big eye, yellowfin tuna, and skipjack. And this is a special distribution of tuna catch in 2023 data. This is a IOTC area, but I mentioned about study areas like this. But we can see very quite dense tuna distributed 15S and 10S very close to coastal area. And this is a visualization and special relationship between tuna catch and acoustic backscatter intensity. So the circle larger is higher, and then the color dark red is in a higher acoustic backscatters. The hollow circle is in tuna catch. So we can visually see, and there is nothing. Unfortunately, data wasn't available in here, so we couldn't have acoustic data. So we try to have some relationship between tuna catch and prey biomass, which is in amphibious. And this is a special distribution of environment variables and tuna catch at 47.4 meter. And here is a temperature, salinity, and DO, and oxygen, and chlorophyll. So we can see temperature peak and salinity in this range, and DO and chlorophyll. And same visualization, but we use the environment data at 130.7 meter. And I just want to mention the tuna catch is from 0 to 300 meter, but we use the same tuna catch, but environment data is two different. One is above threshold, the other is below threshold. And I mentioned about the multi-net and the zooplankton and 3 location and depths and their size and weight and density. And we can see 8S is in the center of SSTR area, is high density in zooplankton. The second highest is 2S, and the below 19S is the lowest density of zooplankton. And we can see dominant zooplankton species categorized by latitude and depths. And 19S and 8S and 20 and 0.0 up to 60 meter and 200 meter and 300 meter. And overall, a 19S is more calanoid and copperboard, and 8S radioladians, and 20 is Pyocystis noctiluca and radioradians. So 3 locations have a different species and different density. And here result of a GAM analysis. And the tuna catch and amphibious, unfortunately not the significant, and the tuna catch and environment and above threshold and below threshold. And we can say GAM analysis tells very weak positive relationship with the prey ability. Sorry, at the 47.4 meter in all environment values was significant, but among them chlorophyll and DO show strongest influence. So tuna catch and is increasing salinity up to 36 and peaked and 27 up to 28 30s. and 830.7 meter in all environments was significant too. And the strongest is the salinity. The DO is the weakest. And tuna catch and rise with the salinity peaked at 18 up to 24 in the southeast. Those are result. And here are conclusion. And this study demonstrated the distribution of tropical tuna species in West Indian Ocean is shaped by variability and the depth-specific environment condition. And tuna catch is highest in equator region with a dense acoustic prey and elevated zooplankton biomass. And the above threshold, above thermocline and 47.4 meter tuna catch increase with a moderate high temperature, intermediate salinity, and elevated DO and chlorophyll, while below thermocline and tuna catch were linked to cooler and saltier waters with a moderate oxygen and chlorophyll. And these depth-dependent patterns highlight how horizontal and vertical gradients influence tuna habitat and prey accessibility, supporting an ecosystem-based fisheries management under changing ocean conditions. Thank you for your kind attention. Thank you.
Okay. Thank you very much, Professor Kang. Totally noted that at March, we can fishing a biggest tuna. This is a joke. Okay, right now we move to the question and answer. For the audience, you can give a raise hand or ask directly. Maybe if you want to ask a question at the chat box, you can to do so. Carry on. Or I'm sorry if I go to bahasa. Untuk bapak-bapak dan ibu-ibu yang mau bertanya dalam bahasa Indonesia, bisa ditulis di chat box atau langsung nanti mungkin kita bisa langsung translasikan untuk Profesor Kang. If anybody wants to ask in Indonesian, you can put it in the chat box or you can ask directly and we can help you to pass it to the Professor Kang. Thank you very much.
Morning, Pak Umar.
Morning.
Is it clear, my voice?
Yes, totally clear. Carry on.
Morning. Okay. Good morning, Prof. Kang. Thank
Hello.
you for your presentation. My name is Iis Sri Yulianti. I'm from Deep Sea Research Center. I want asking just one. As we know that we are facing the climate change, and we know we sense the impact of the climate change, like the ocean, the oxygenation, the increasing of SSO, and the decreasing of pH. Is there is the impact of those conditions to the tuna catch or the tuna production, especially in the western of Indian Ocean.
For me, this is first time to
Thank
use.
you, Kang.
Thank you for important question. Thank you so much. And it was first time for us, our team to use tuna catch, but there are a lot of people already in the papers. They tuna catch based on IOD. IOD, this is index. This is really directly connect to the climate change. This is one index. Of course, IOD is based on the sea surface temperature, but there are a lot of environment, too. But many people, like say IOD, how affect, tuna, species and their biomass and their, distribution. And I don't exactly remember. I cannot, show, which in a paper and which in a title. But when I, publish, this work, and I read in many papers, that is my memory. And there are already, some studied connection between climate change and tuna biomass and tuna distribution.
So we can say that also impact the production of tuna, yes?
Like
Well, the condition is
like
I think.
Like thinking about this one is in a positive in IOD event, and that is in a negative IOD event. So the west side, the tuna and specifically yellowfin tuna catch more in IOD event. So this is just one example, but I cannot tell like climate change, like IOD events, represent entire tuna catch. But IOD is affected by climate change, so that we can use one example, so tuna catch or some other elements affected by IOD, and which is linked to climate change.
Okay. Thank you, Prof. Kang.
Thank
Silakan,
you.
Pak. Pak Omar, saya berikan ke Pak Omar.
Okay, thank you very much for your answer, Professor Kang. I was ask something as a geophysical myself. Maybe before you said that use frequency differencing or delta MVBS to classify acoustic scatters like swim bladder fish versus crustaceans. Since the acoustic impedance of both the seawater and the biological targets change significantly with depth, temperature, and salinity, especially between the two depths you analyze. I believe is 47.4 meters and 130.7 meters. My question is, how sensitive is the frequency differencing method to the changing geophysical properties of the water column? Maybe only that. Thank you very much.
Thank you for interesting question. And before we decide, two depths, one is above thermocline, the other is below thermocline. And in those area, and thermocline is between just above 100 meters, sometimes shallow, sometimes deeper. So that this depends on many effect. So the ideal is like temperature. As you know, in the fish is a barrier. They cannot go. They cannot get through. Their like not optimal. They're durable in a temperature. So one like say many people talking about the temperature like the even thermocline is changing based on many elements. And so I believe, and so we this is just one snapshot. This is very short time, and but like this is like 2 months time 2023, and then one above threshold, one below threshold. But I believe like say fish like tuna, and they move around and based on thermocline. So I cannot tell right now tuna like for long term I haven't done long term monitoring, or I haven't studied long term. I haven't used long term data, but I believe tuna distribution and their catch is really based on thermocline or based on the temperature and other the environment variables. Is it? Yeah. Is it answering your question?
Yes. I believe we have to prolonging the survey. I believe if we have the data for 2024 or 2025, maybe this will answer the the question itself. But I see the pattern here, and I believe you. The answer is the same as your answer before. Okay, move on. There is from Widya Nugroho, I believe there's a question from chat box. The question is, thank you, Professor Kang. Thank you for your excellence and provide a thorough verification of tuna catches and the marine environment. However, a few aspects remain unclear. What specific fishing gears were used, whether long lines, hand lines, or cruise signs. Did the fishers utility fetch rumpon, fish acquisition device? If so, were drifting or anchor fetch? Additionally, could the findings of your study serve as a reference or basis for recommendation, such as mapping potential fishing zones, PFZ, or determining the most strategic location for fetch placement? Vidya, Print from Hokudai. Okay, the floor is yours, Professor Kang.
Yeah, I hope I understand your question well. Yeah, but the IOTC, there are variety of the data source. So what we have is the data, like say this is a data reason, this is obligation. Like fishermen, they have to report. So it depends on the fishing gear, and sometimes 1 degree, at least one data source. Sometimes 5 degree, one data source. So data has a year, month, and fishing method, and then location, latitude, longitude, and then body length range. Not one specific the body length, exact body length, so that we got a median value, and then count. So yeah, I mentioned about the fishing method, and sometimes they do percent, they do gillet, they do long line, and then they report. This is a like logbook, and I'm long line, and year, month, and location, and then the size and how many. That's it. So that that is what we have. So that we have to convert based on personal species, but based on gear type. And if they say, okay, gillet, okay, and then we use this equation, and that I mentioned about, we know L, the body length, but we don't know weight, the round weight. So that the data is this species and this gillet, this gear type, and then using this equation, and then convert the weight, something like that, so that we kind of add up and based on species, so that we the visualize in the data. Is it enough?
Okay, I believe there's no chat box anymore. I believe that's maybe answer the cache. I hope that maybe answer the question. If okay, Miss Widya is asking again, pardon, another question if possible. Do you include depth contours in your model? For example, features like seamounts or knolls can create upwelling or current change that might help verify results. It's interesting about the hydrographical aspect of the
seamount and what current?
knolls. Is kind of like little seamounts.
Okay, seamount. And
that can create upwelling or current changes that might help verify your results. It's maybe hydrographical aspects.
Yeah, but those are in open sea. I 100% support the sea mountain or sea hill. The hydrographic information is very important. And I haven't done what is it, like eddy or any current and any like sea height information. But I know many people they do Madagascar and those in area in a sea mountain, and then they linked the hydrographic information within a tuna or within some other animals. But here it's very open sea, and the depth is like 30,000 meter, and then more than 30,000 meter. Actually, we had mooring here. It's more than like 30,500 meter. So it is open sea. Of course, when the vessel voyage start to is it Mauritius, and this is land. This is very close to coastal area. But we normally look at this in line 65 in E line is open sea, and yeah, I believe, and then it's very deep. So uh, very deep, and then tuna, and the major distributed depth is up to 300 meter. So that we took the 300 meter in acoustic data and tuna catch environment data, and yeah, but your question, I don't think it affects, or it is connected to this area. It should go more like coastal area. But I believe the sea mountain of course is very important, and like eddy important, but future no sea mountain there. Of course sea mountain is in really deep sea, and but I'm very interested in eddy and within some other macronecton or in other animal. Thank you.
Okay, excellent answer. Because I believe the tuna itself is come from the open sea. So the hydrographic also affect them, maybe with the bigger features like seamount or something like that. Uh, let's see. Is there any question? You can raise hand or you can ask in Indonesian. Anda bisa mengirim dalam bahasa Indonesia. We have 3 minutes to go to ask question. Okay, okay. There is another question from Mr. Fuad and Emira Nabila. First from the Mr. Fuad. Thanks for interesting question, Professor Kang. You have mentioned about SCTR upwelling region that might influence marine primary production. Could you explain what mechanism drives this upwelling region?
Yeah, so I cannot tell mechanism, but many people, this is I normally use two papers, and the arrow I didn't put the detail, but this is current in Alpha-Gun, like their full name, so that those in a current makes upwelling. So yeah, definitely in current, and then that's why this slide, this is a associated area, yeah, based on this temperature, and then this chlorophyll, and then like some average nitrate. And what I'm saying is yeah, it obviously very different. It's very cool, and then very high chlorophyll, And so that this is open sea. It's not really close to coastal area. So this is a very unique feature, and that upwelling bring the nutrition for phytoplankton, and the phytoplankton, and then zooplankton, and those the color and shows, and it is a upwelling area, cooler, much cooler, and then very high in chlorophyll and very productive in area. And I don't know all the mechanism, but as far as I know is the current, and of course current is connect to the air, the atmosphere and the wind, so that it varies. So that sometimes the SSTR area is we say this is a rough area, and center we believe in ADS in area, but it is moving. It's not constantly same. So each year or other elements in the make its area sometimes expand, sometimes shrink, sometimes very shallow. It's very cold, or sometimes mild. It's very various. But as far as I know, this is very productive in area compared to other, especially like southern part of Indian Ocean. Yep. Thank you.
Okay, maybe one more question from Emira Nabila. Thank you for your excellent presentation and providing a comprehensive verification of tuna catches and their relationship with the marine environment. However, I would ask about the data collection process, technical part. Were there a particular challenge or difficulties encountered during the data collection? If so, did you observe any anomalous data or unusual patterns while collecting the acoustic data? Maybe it's a technical part. It's interesting.
Like say this is very remote area, and then not many people come to here, and even like those especially India, they spend some time, of course, that research, which means budget, research budget, so that it is non-studied area. And Korean, we have to go there, but this is a kind of a month survey, which is very expensive. And not only acoustic, there are a lot of gears, gliders, and I mentioned about heelys and mooring, and then they do algo buoy, and then they do I forgot all the they of course they do CTD and a lot of things. So, and very limited in a time, and of course you better have not only the multi-net and other the gear like 20. So I mentioned about, oops, sorry. So those 2020, 21, and we had a little bit the big net, 2 meter, and large mesh net. So it's better to have a multiple net and sampled various the organisms, but again is a limited in time and limited in a budget. It is very difficult. And the tuna, luckily IOTC has a well organized, but it doesn't mean in a very perfect. There are some way you have to understand and then see, and but still available, better than nothing. And the one is this is just too simple. Acoustic is in 6.5 in E, and we need more coverage and acoustic data specifically to tell and direct relationship between tuna catch and the MBBS, which is prey biomass, or acoustic backscatter. But that is what we have, and this is a way to show the method integrated in a methodology, so you can use acoustic, which is micronecton and that plankton, And then if you have, like phytoplankton, and then you can have, some relationship, and you can have, like upper in higher tropic, which is in a tuna. And then you may have, broad idea about, eco-ecosystem. But again, this is very expensive, research. But like 3 point is not enough, and acoustic line is not enough. But this is, what we have, and this is what we can, do. But hopefully, and we will have, more, sample station and, broad, acoustic, coverage in the future. Thank you for interesting question.
Okay, thank you for interesting insight and thank you for the interesting answer. I'm very much say that thank you very much for the answer. And then the time is up. I'm real sorry to hear that. But Professor Kang, I would like to take this opportunity to sincerely thank you once again for your valuable time today and sharing your excellent research and insight. But I fully understand, we fully understand that you have another commitment to attend. Please feel free to the session whenever you need to. We truly appreciate you taking the time with us today. And I hope there will be many more opportunities for collaboration and discussion in the future. Thank you very much, Professor Kang. And in Korea, I believe they say 감사합니다.
감사합니다.
Have a safe and a wonderful day.
You too. Thank you. Thank you for
very much.
inviting me. Thank you. Thank you.
Okay. Ladies and gentlemen, thank you once again to Professor Kang for that insightful presentation and discussion. We will now move on to our second presentation. I believe if there is no another global guest from our presentation today, I will change to bahasa, if that's okay. Sekarang kita akan masuk ke Dr. Manulang. Dr. Manulang, mungkin kita bisa mulai presentasinya, jika memungkinkan.
Okay. Can I share my share screen? Can I share my screen? Oke deh. Oh, thank you. Sudah terlihat, Mas Omar?
Sudah terlihat dengan baik, Mbak. Silakan, the floor is yours.
Oke, thank you. Oke, karena aku ini pakai internet, ini mungkin aku bisa ini dulu kali ya. Okay. Maybe my presentation, because I already prepare in English and to practice my English. So can I present in English, Mas Umar?
Of course you can. Excellent.
Okay, thank you. Maybe we can combine bahasa Inggris, bahasa and English, right? Okay. Okay. Sorry. Okay. Good morning, everyone. I'm Koryanti Manulang from Research Center for Deep Sea, BRIN. Thank you for having me here today. Today, I would like to share what we have learned from several studies along the Indonesian through flow, following microplastic from the water column, the copepods, and eventually to the deep sea sediments. And this is not the result of one study alone. It brings together several collaborative studies and publications involving colleagues from BRIN, FIO in China, University of Maryland, USA, IPB University, and University of Indonesia. So let me begin with the simple question. Where does plastic go when it disappears from our sights? To answer that, we need to stop thinking that the ocean is only a surface and start looking through the entire water column. Microplastic can move because it has a very tiny, so microplastic can move with current, remain within particular water layers, can also ingested by marine organisms, or eventually reaching the deep sea sediments. In these presentations, I will look at this journey from the four perspectives. First is ITF circulation, the water column, biological uptake, and deep sea sediments. This perspective come from different studies, but together they give us a broader picture where microplastic occur and which process may shape their distribution below the surface. So first, let we begin with what is the Indonesian true flow. The Indonesian true flow, or normally we can say ITF or Arlindo, is a major ocean current system that carries water from Pacific Ocean through the Indonesian seas and go toward to the Indian Oceans. You can think the Arlindo or ITF like a river in the oceans, except that has no visible banks. Along the way, the water moved through the narrow straits, and the main pathway is the Makassar Straits is the main pathway of the Indonesian true flow, and it is the deep basin. And the ATF carries much more than water. It's not only the water, but also salt, nutrients, organism, and particles, and maybe microplastic. Microplastic can also become a part of the moving systems. Of course, besides from the Pacific, the microplastic also can enter the ocean from the local sources within the Indonesian. But once they are in the sea, ocean circulation can influence where they travel and how they distribute it. This led us to simply question, how can we follow microplastic beyond the surface and through the three dimension of the oceans? So to answer this question, we need to look at down through the water column and sample different part of the system. And so how we follow the microplastic of through this three-dimensional ocean. From our studies, we use different sampling approach to look at different part of the system. The first, we use here the rosette, the bottles, rosette bottle CTD, Niskin bottle attached with the CTD. And from our study, we sample the water from the 5 meter until 2,550 meters. We also collected copy pouch using the plankton net. We throw the water with the plankton net from the 300 meters, and from the deep sea sediment we use the box score. So instead of looking only across the ocean surface, we began to look down through the ocean, and this led us to our first result here. That from our study, we have already seen that microplastic were found from shallow water down to the more than 2 kilometers. So we found microplastic in the 5 meters until the 2,550 to more than 2 kilometers. But when we look more closely, their concentration varied across different depths. Some depths show higher concentration than others, suggesting that microplastic do not simply move downward in a uniform way. So the important question was no longer simply, are microplastic present below the surface? It's not. The next question became, what controls? They remain within the water column. And one important feature that emerged from our analysis is the thermocline. So, what is the thermocline? Thermocline is the layer where water temperature changes rapidly with depth. As a temperature change, water density also changes, creating different layer within the water columns. From our study, interestingly, many of microplastic we found in the thermocline and near the thermocline or below the thermocline. For very small particles such as microplastic, this phase structure can influence whether they continue sinking, remain suspended, or move with the surrounded water. In our study, the temperature and density were the environmental factor most strongly associated with the microplastic concentration. So here, if we look at in this graph, here this is the thermocline. We found a microplastic here, more microplastic in the thermocline. But it does suggest that microplastic do not simply fall straight from the surface to the seafloor. Their movement can be influenced by the physical structure like water column. And if the particle remains suspended in the water, where many marine organisms live there. So the next question is, are marine organisms already ingesting the microplastic? So from our study, we have found that microplastic can ingest it by the marine organism, even the tiny marine organism. From our study, we use the copepods. Copepods are the tiny crustaceans, but they play a very important role in the marine food web. They feed on smaller organisms and in turn become food for the fish, the bigger fish or other marine animals. Along the main pathway from the Makassar Strait, we examined about 6,000 copepods and found 133 microplastic inside them. So on average, this is about 1 particles for every 55 copepods. Maybe we can say that that number sounds small, but finding plastic inside the organism collected from the natural marine environment tell us that exposure is already occurring. Most of the particles were fibers, about 87%. Fibers were also dominant in the water columns. We examined 3 different copepod size groups. Large copepods show slightly higher ingestion rates, but the difference among the groups were not statistically significant. But we found the bigger copepods, we found the more microplastic, and the smaller copepods, we found the smaller the microplastic number. So along the main ITF pathway, we can see how the movement of the microplastic through the water column can eventually become exposure in marine life. But this is only one part of the much larger system. The Indonesian to flow. Here from the first study from the water column and the copepods, the study were conducted in the main pathway of ITF in the Makassar Strait. But for the next slide, we will see how the microplastic behavior in the different side of the ITF pathway. So Banggai C is the still is a how to say is include from the ITF bagian dari ITF, but in the eastern part. Okay, so after looking the main western pathway, now we want to know what happened in Banggai Sea. So this region, so Banggai Sea lies along the eastern route where water from Pacific Ocean entered through the Maluku Channel. But the Banggai Sea is not simply another point along the current. It's a semi-enclosed basin that also experience seasonal upwelling. So from the third study, we're looking especially for how microplastic behavior during the upwelling season. So upwelling brings deeper, cooler water toward the upper ocean. This creates a vertical movement within water column that already contains microplastic. That makes at the Banggai Sea an interesting natural setting for asking a new question. What happened to microplastic when the ocean water moves not only horizontally, but also vertically? We collected water from the 22 stations, starting near the surface and extending depth of approximately about 1,000 meters. So, and the time from the research cruise from the study is the during of the expected of the upwelling. So, and when we examine the vertical pattern, the one familiar layer appear again. Here the thermocline. So we found again, okay, microplastic high again in the thermocline. And but when we compare the average concentration among the four water layer, even the thermocline were higher than others layer, but the statistically is not significantly. So that's why we need to be more careful with the interpretation. So instead that this result tell us what the thermocline may be still an important part of the story, but it is probably not the only process controlling the distribution microplastic, because the study is conducted during the expected upwelling. So we do analysis from the how how much the microplastic in the station with the affected with the upwelling, and how much the microplastic concentration with the station not affected with the upwelling. So when we separate, after we separate the upwelling station and non-upwelling station, we found something interesting. At the thermocline, the average concentration was lower in the upwelling areas. It's about 20 particles per liter compared to the non-upwelling areas in the 30 particles per area. So we found a lot more microplastic in the non-upwelling areas. Upwelling increased vertical water's movement and mixing. This may reduce the retention of microplastic within one particular layer and redistribute them across the wider depth range. So rather than simply concentrating particles, upwelling may help spread them vertically through the water column. This reminds us that microplastic distribution is not controlled by depth alone. It is also shaped by how the ocean moves. And if some of these particles continue moving downward, what happens? Do they eventually reach the deep sea floor? So this is the next part from our study. We have followed the microplastic through the water column from the surface through the thermocline and into the deeper water. So, and the study is still conducted in the Banggai Sea. In the Banggai Sea, we collected sediment from 5 stations using a box score. The stations were located at depth between approximately 1,600 until 2,450 meters. And we found microplastic in all the stations, the 5 stations. Their abundance ranged from 200 until 800 particles per kilogram, with an average about 533 particles per gram. Once again, same with the 3 previous study, that fibers were the dominant shape, accounting for 92% of all the particles we found. This is interesting because fiber were also common in our if we look at from the review from the even from the Indonesian region or the global, is always fiber is the most dominant microplastic shape in the marine environment. So but it doesn't mean that we track the same fibers, all the way from the surface to the bottom, but it shows the consistent pattern across the very different parts of the marine environment, even in Indonesia or regional or global. We often imagine that deep sea as a remote and protected from human activities, but these findings remind us that the seafloor is connected to the everything happening above. Right? So, however, the seafloor may not always be the final resting place where particles accumulate can also depends on distance from the coast and seafloor, topography, and ocean circulation. So, from the this is the result from the microplastic from the deep sea sediment. We found the closer to the coast, the more microplastic we found. Actually, if we compare to other study, sometimes commonly they found like the deeper of the seafloor, the more microplastic we found. But in this study, we found another perspective, that the closer to the coast, the more microplastic we found. So in our study in coastal station, we the closer station about 20 kilometers from shore, where the more offshore station is about 80. So we here the station 5, station 2, station 1, it's about 20 kilometers from the coast, and here station 3, station 4 is about 50 until to 80 kilometers. And statistically, we found a strong negative relation between distance from the coast and microplastic abundance. In simple terms, the farther we move the coast, the fewer microplastic we generally found in the sediments. This pattern suggests that local sources are important. Even like a common assumption that deep sea area is a sink for microplastic, but the source, like local source, is important. We also found similar types of particles at offshore station. This suggests that regional ocean circulation, including the Indonesian through flow, may help redistribute microplastic beyond their original sources area. So we may see the process operating together. Coastal activities introduce particles, while current and seafloor process influence where particles are transported and eventually accumulated. So we almost reached the last slides. So what do this for study tell us were put them together? They show us that microplastic do not follow one simple path through the ocean. Current can transport them. The thermocline can influence where they remain, and upwelling can redistribute them across different depths. While suspended in the water column, they can also interact with the marine organism, as we saw in the copepods in the second study. And eventually, some particles reach deep sea sediment. Of course, we did not track the same because it's from the to location, different location, but they show that the ocean is connected from the three-dimensional system. Microplastic can move, remain suspended, be ingested, settle, and possibly be suspended again. So when plastic disappears from our sites, it may simply have moved to another part of the ocean. And this leads us to important point. If we monitor only surface, we may miss something. And like, okay. And so what doesn't mean, what does this mean for the way we study microplastic pollution? Surface sampling is important, but it gives us only the one part of the picture. If we look only in the surface, we may miss particles around the thermocline, in deeper water, instead marine organisms and deep sea sediments. We also need to understand how the ocean moves. Temperature, water density, currents, and upwelling are not only background conditions. They help us to explain where microplastics are found. So future monitoring should be connect different depths and different parts of the marine environment, from water and marine life in the seafloor. Simply put, a three-dimensional problem needs to three-dimensional monitoring. So before I close, I would like to leave you with three simple messages. First, microplastics are not limited to the ocean surface. They are also present in the deeper water and deep sea sediments. Second, in the ocean itself, where they are found. Current, water layer, upwelling can transport and redistribute them. And third, this movement is more than physical transport. It also leads to exposure, marine organism, and accumulated in the deep sea floor. So even the ocean surface look clean, we may see only one part of the story. To understand plastic pollution, we need to look the occur not only across it. Okay, thank you, and I'm looking forward for our discussion.
Okay, thank you very much, Dr. Cory. Excellent presentation as always. Sekarang kita masuk ke sesi diskusi, sesi tanya jawab. Dipersilakan kepada rekan-rekan sekalian untuk mengajukan pertanyaan bisa melalui raise hand atau bisa kita sampaikan langsung dalam bahasa Indonesia atau bagaimana. Dipersilakan kepada teman-teman semua untuk mengajukan pertanyaan. Waktu dan tempat saya kembalikan lagi ke penanya. Ya, untuk Mbak Is dipersilakan, bisa langsung saja.
Baik, terima kasih Mas Omar. Terima kasih Mbak Cory atas presentasinya. Izin saya bertanya lagi ya. Dari paparan yang sangat menarik yang sudah disampaikan Mbak Cory. Berarti kalau tolong dikoreksi kalau saya salah ya. Dalam persebarannya dari surface sampai bottom, sampai dasar laut dalam, untuk mikroplastik, faktor yang paling mempengaruhinya adalah apakah berat jenis dari jenis mikroplastiknya atau ukuran dari mikroplastiknya atau faktor fisika oceanografi? Mana yang mungkin lebih kuat dalam mengatur atau driver utama dari persebaran mikroplastik tersebut? Itu saja dulu Mbak Kori untuk memancing yang lain. Silakan saya kembalikan ke Mas Omar.
Ya, mungkin bisa langsung didiskusikan.
Oke. Makasih Bu Is. Jadi sebenarnya kalau kita belajar tentang apa ya, jadi vertikal tentang bagaimana sih mikroplastik itu bisa settle down ya, dia bisa turun ke bawah, itu sebenarnya ada banyak hal gitu. Jadi pertama kalau misalnya dari studi kami itu tuh dipengaruhi tadi misalnya kalau physical oceanography mempengaruhi ya karena ketika dia terbentuk lapisan termoklin. Kalau misalnya terbentuk lapisan termoklin kan dia misalnya lapisannya itu kayak lebih rigid gitu ya. Jadi kayak mungkin mikroplastiknya jadi kayak lebih stay di situ karena dia nggak bisa ke atas nggak bisa ke bawah gitu. Nah tapi selain itu sebenarnya ada banyak hal gitu kan. Jadi mikroplastik bisa turun ke bawah itu selain fisikal juga ada juga dia bisa eh namanya marine snow. Jadi kalau misalnya di laut itu kan ada marine snow. Jadi microplastik itu kan karena dia tiny gitu kan ya, kecil dia bisa kayak eh, ke apa sih dia menempel ke marine snow dan bisa jadi ke bawah. Bisa juga dia ikut misalnya dia dimakan sama hewan, terus hewannya misalnya pup dia di water column terus dia nanti pupnya jatuh dia ikut jatuh gitu kan jadi atau juga dia ada proses yang namanya biofouling jadi dia menempel ke biofouling gitu jadi berat jenis ya tentu saja berat jenis itu seperti studi saya yang pertama itu sebenarnya saya juga menghitung kecepatan turun dari setiap partikel yang saya temukan. Jadi itu, oh karena misalnya dia PP, dia berat jenisnya berapa, jadi dia mungkin karena berat jenisnya lebih besar, jadi dia bagaimana dia dibandingkan ke berat jenis air laut, gitu. Jadi dia bisa lebih cepat turun gitu. Jadi sebenarnya ada banyak hal, Mbak. Makasih.
Oke, baik, terima kasih.
Oke, selanjutnya ada pertanyaan menarik nih dari Pak Adi Samudriadi dari BRIN. Hello Dr. Cory, thank you for your interesting presentation. Could you explain your finding from sediment sample? Thank you very much. Jadi yang ditanya ke sedimen ya, jadi bagaimana mikroplastik ini ada di sedimen. Mungkin bisa dilanjutkan diskusinya, Mbak Cory.
Oke. Jadi jadi kalau misalnya dari hasil sedimen ini, makasih Pak Adi. Jadi kalau dilihat dari sedimen ini bisa kita lihat bahwasanya uh, lebih banyak mikroplastik makin banyak mikroplastik yang ditemukan karena kita sampai menemukan 200 sampai 800 mikroplastik di dalam sedimen gitu. Jadi explain ya finding sediment sample. Oh, bagaimana ya finding from the sediment sample. Jadi
Iya, finding ya kayaknya.
Oh, ini pakai box score, Pak. Mungkin ini Pak Adi juga membantu saya nih mengambil sampelnya nih. Kita pakai box score, Pak.
Oke,
Gitu.
siap.
Gimana, Pak?
Ya, mungkin profil mikroplastiknya ya dari sedimen seperti apa gitu, Bu Kori?
Oh, profilnya gitu ya. Oh, jadi sebenarnya eh, karena waktunya terbatas ya, jadi saya nggak sampaikan. Jadi di samplingnya ini tuh kita bagi tiga sebenarnya. Jadi kita yang surface, yang tengah, sama yang di dalam gitu ya. Jadi kayak per 5 cm gitu ya, Pak ya. Nah, kita menemukan mikroplastik paling banyak ditemukan tuh justru di tengah, Pak. Gitu. Jadi mungkin karena proses juga apa kadang-kadang kan oh kenapa dia nggak di surface-nya gitu kan. Mungkin karena proses juga upwelling gitu ya Pak ya. Jadi makanya yang paling banyak itu justru di tengah-tengah gitu Pak.
Menarik ya, mungkin bisa dikonfirmasi dengan data sedimen core ya. Bisa lihat nanti apa variasi secara vertikal mungkin ya ke depannya ya.
Iya, Pak makanya kayaknya mungkin kita harus lebih banyak pelayaran ya Pak ya untuk yang bisa
Oke,
mengambil sampel
siap.
mikroplastik.
Siap. Premium sudah dibuka ini. Welcome. Nanti apa sampel-sampel sedimen mungkin ya di
Iya,
banyak area di Indonesia bisa kolaborasi lah.
iya Pak, makasih Pak.
Sukses, Bu Kori.
Makasih Pak, sukses juga Pak Adi.
Terima kasih Pak Adi untuk pertanyaannya. Lanjut, ini ada pertanyaan nih dari Nurfianto Giar Pangindung. Selamat siang, saya ingin bertanya Bu Kori, ikan pelagis apa yang bioakumulator dan biomagnifikasi yang terbesar dan banyak dikonsumsi masyarakat sehingga menjadi warning bagi nelayan dan konsumen? Waduh, ini pertanyaan bagus sekali nih. Jangan makan ikan ini gitu. Ayo mungkin bisa ada insight-insight Bu Kori,
Iya.
ikan apa nih yang perlu dihindari nih kalau
Iya.
makan mikroplastik banyak? Terima kasih.
Iya. Jadi tuh sebenarnya Pak ya, makasih Pak Nurfianto pertanyaannya. Jadi sebenarnya studi mikroplastik dengan logam berat itu sedikit berbeda gitu ya. Jadi kalau misalnya logam berat kan kita bisa tracking nih ya gitu kan ya, karena dia kan bisa ketika misalnya dia sekali dia makan misalnya dia makan polutan dia akan stay ya. Jadi kita bisa lihat di livernya, kita lihat di atau hatinya gitu kan. Jadi kita bisa oh yang ini nih, kalau misalnya kayak kayak untuk studi logam berat misalnya kayak tuna itu kan sebenarnya kan kayak ada warning juga ya kalau misalnya oh dia bisa ini dan semakin lama. Nah, untuk studi mikroplastik sebenarnya Pak, jadi mungkin karena juga studinya ini masih masih belum banyak terutama untuk yang pelagis ya Pak ya. Jadi rata-rata biarpun meskipun ada di ikan gitu ya, di ikan itu masih yang coastal gitu. Jadi kita belum bisa pastikan sebenarnya kalau yang di hewan pelagis bioakumulator itu bagaimana gitu. Kalau saya lihat-lihat dari review itu masih sedikit gitu kan. Paling yang banyak itu tuh masih di tuna aja. Tapi kan kita belum bisa mengatakan oh tuna paling besar gitu kan karena belum apa ya, belum banyak studinya. Dan ini kan sistemnya kalau dia makan mikroplastik dia dikeluarkan gitu ya dari saluran pencernaannya juga begitu gitu Pak. Jadi mungkin ke depan sih kalau saya itu eh, baca-baca sebenarnya kalau misalnya kayak di luar negeri itu tuh studi mikroplastik itu tuh nggak hanya ini lagi. Oh, kita menemukan berapa mikroplastik di tuna, berapa mikroplastik di kopepet, bukan lagi. Tapi kita melihat bagaimana sih ketika mikroplastiknya banyak, bagaimana sistem sarafnya, bagaimana sistem reproduksinya, karena dia memang dia apa ya, ternyata kan terdampak gitu kan. Jadi itu sih. Mudah-mudahan sih makin ke depan kita bisa buat studi-studi seperti itu. Jadi kita bisa melihat DNA-nya, kita bisa melihat proteinnya seperti apa. Apakah dengan banyaknya mikroplastik, misalnya, oh di timur nih banyak mikroplastik, atau apakah memang proteinnya di timur dengan di Indonesia apakah berbeda atau enggak gitu. Gitu sih Pak. Semoga ini ya Pak ya.
Oke,
Semoga.
terima kasih Dr. Kori. Mungkin pertanyaan dari saya ya. Jadi dasar laut kita itu udah resmi menjadi tempat sampah mikroplastik terakhir yang bakal permanen atau mikroplastik ini ada potensi naik lagi ke permukaan?
Iya.
Atau dilihat dari tadi dari grafiknya coastal itu kan jadi tempat yang banyak numpuk gitu, atau dia bakal mungkin jadi naik lagi udah gitu pergi ke tempat lain. Mungkin itu aja kan.
Iya, makasih Mas Umar. Jadi kalau ngelihat dari semua studi yang dilakukan di laut dalam, pasti banyak mikroplastik ya di laut dalam. Bahkan yang bilang, oh semakin dalam semakin banyak mikroplastik. Seperti kan mikroplastik udah ditemukan juga di Mariana Trench ya. Jadi dan di kayak kedalaman di atas 6.800 itu makin banyak mikroplastiknya. Dan kemungkinan untuk dia bisa naik ke permukaan lagi yaitu kalau ada apa ya upwelling itu yaitu gerakan dari gerak arus dari bawah ke atas itu sih atau kita bersama-sama mendrawing ya narik dari bawah
Ya
gitu.
berarti masih ada kemungkinan dia bakal naik lagi dan
Mm-mm.
dimakan oleh ikan-ikan lagi ya. Ini menarik sekali. Oke, mungkin untuk peserta yang lain masih ada, kita masih ada waktu 2 menit lagi sebelum saya melakukan closing remarks, dipersilakan.
Ada dari Pak Adi lagi.
Oke, mungkin ada dari Pak Adi ya. Ini lokasi studi ini tidak ada oceanographic mooring. Jadi untuk jalur ITF yang ada mooringnya bisa juga menggunakan barnacle atau tritip untuk akses mikroplastik bioaccumulation. Oh ya, betul, betul,
Iya.
betul.
Iya benar Pak Adi. Jadi sebenarnya tapi ini bukan yang studi yang ini ya Pak. Tapi yang studi berikutnya kemarin tuh apa sih yang dengan IOKAS sudah mulai dilihat dan memang benar mikroplastik banyak ditemukan juga di bernakelsnya. Tuh, jadi ya ini sih memang. Jadi memang sih karena studi laut dalam itu mahal ya. Jadi kita itu benar-benar kayak mencoba-coba dan juga kalau tidak salah yang studi yang di Selat Makassar itu masih kayak awal-awal banget. Jadi kita masih mencoba-coba gimana sih kita mau mengeksplor gitu mikroplastik gitu. Jadi tapi ke depan ini tuh kayak makin berkembang. Makanya oh mungkin kita bisa lihat ya dari muringnya gitu. Dan kalau misalnya saya juga pengen lihat itu tuh sebenarnya yang paling ini adalah kayak ininya juga hewan-hewan yang di dasar laut, bentik-bentiknya itu juga yang belum kita lakukan gitu. Mudah-mudahan ke depan bisa kita lakukan.
Oke, mungkin dari chat box ada lagi nih pertanyaan terakhir mungkin ya. Izin Ibu Kori berarti untuk jenis ikan yang lebih berpotensi besar terhadap MPS adalah demersal, pelagis, dan lain-lain. Terima kasih Bu.
Makasih Bu Erpina. Jadi marine organism. Jadi apa ikan itu kan bisa kenapa dia bisa terkena mikroplastik karena jadi dia kan tidak punya kemampuan menganalisis oh ini makanan saya, ini bukan makanan saya. Jadi ketika lewat di depannya udah makan aja gitu kan. Jadi sebenarnya mana yang lebih berdampak sebenarnya ya lingkungannya dulu yang mana yang paling banyak mikroplastiknya gitu kan. Jadi kalau memang tapi rata-rata memang karena coastal itu kayak karena lebih dekat sumber gitu ya. Jadi lebih banyak mikroplastiknya. Jadi eh, kemungkinan gitu. Tapi ini bukan selalu ini pasti ya gitu kan. Jadi kalau karena mikroplastik lebih banyak di coastal gitu kan. Jadi ikan demersal gitu. Tapi kalau tapi bisa juga hal yang lain gitu. Kalau misalnya perairannya bersih banget gitu ya berarti dia nggak kena mikroplastik karena dia kan jadi ikan kayak copepods atau ikan tuh dia itu kan makan jadi seperti copepods gitu kan dia kan makanan aslinya itu filamen gitu kan filamen yang kayak benang-benang gitu kan ya berwarna-warna. Nah mikroplastik juga gitu dia seperti filamen kan benang-benang berwarna biru merah ya udah dianggapnya makanannya gitu, gitu sih. Makasih Bu.
Oke, saya lihat sudah cukup dan waktunya udah 13 menit lagi. Kita bisa closing remarks ya. Terima kasih untuk semuanya. Terima kasih Mbak Kori untuk presentasinya. Ini presentasi yang bagus banget. Kita udah sampai ke penghujung forum hari ini. Ya, saya ingin melakukan waktu sejenak ya untuk mengulas kembali presentasi dan diskusi yang menarik banget ini yang kita ikuti hari ini. Jadi kedua presentasi ini memberi dua perspektif yang berbeda ya, namun saling melengkapi dalam baik kita memahami laut dalam terutama. Yang pertama itu presentasi dari Profesor Kang menunjukkan pentingnya integrasi observasi akustik, geologi, dan lingkungan untuk memahami sebaran perilaku tuna tropis. Salah satu pesan utama kritik yaitu organisme laut itu nggak bisa merespon pada satu faktor lingkungan. Jadi interaksi kompleks antara ketersediaan biologis dan kondisi lingkungan yang bergantung pada kedalaman air juga. Jadi temuan yang perbedaan kontrol lingkungan itu mengingatkan kita pentingnya melakukan observasi lautan juga dalam ranah 3 dimensi. Lalu dari Dr. Kori Yanti Manulang yang mengarahkan perhatian kita pada dimensi penting yaitu mikroplastik yang langsung berpengaruh kepada hidup kita sehari-hari. Jadi presentasi beliau tuh menyoroti bagaimana proses-proses oceanografi air lindo yang mempengaruhi pergerakan sebaran mikroplastik dan proses-proses biologis lainnya, yaitu mengakumulasi pada lingkungan laut dalam dan coastal juga. Jadi hal ini menunjukkan mengapa ilmu kelautan laut dalam tidak dapat ditangani hanya melalui satu disiplin ilmu dan satu observasi saja. Bagi kita umumnya researcher, pendekatan multidisiplin ini menjadi sangat penting sebagai negara kepulauan yang berada di persimpangan laut utama, yaitu ahli itu sendiri. Saya percaya bahwa forum seperti ini menyediakan wadah yang penting untuk merintis koneksi-koneksi tersebut. Pengetahuan yang dibagikan bisa menjadi titik tolak bagi pertanyaan riset yang baru. Apa yang akan kita lakukan, apa ke depan yang bisa kita lakukan, dan apa yang akan kita riset baru. Atas nama Pusat Riset Laut dalam BRIN, saya ingin menyampaikan apresiasi yang mendalam kepada Profesor Mi Hui Kang yang telah berbagi riset dan keahlian yang sangat berharga bagi KRI ini, dan Dr. Koryanti Manulang atas presentasi dan wawasan yang luar biasa mengenai mikroplastik dan Arlindo itu sendiri. Kami juga mengucapkan terima kasih kepada Profesor Aan atas kata sambutannya, penyelenggara, dan tentunya seluruh peserta yang hadir dalam berkontribusi dalam diskusi hari ini. Dan untuk seluruh peserta, saya harap Anda tidak hanya membawa pulang pengetahuan ilmiah yang baru dari forum hari ini, tapi juga ide-ide dan peluang baru untuk berkolaborasi seiring dengan berlanjutnya perjalanan kita menuju pemahaman dalam. Sekali lagi, terima kasih banyak kepada seluruh pembicara, peserta, dan rekan-rekan sekalian. Menutup forum hari ini, punya satu pesan harga, memahami laut dalam menuntut kita untuk melihat melampaui batas disiplin masing-masing dan memandang lautan sebagai satu sistem yang sangat terbuka. Yang ini saya resmi menutup Dipsi Science Forum hari ini. Terima kasih atas waktunya. Assalamualaikum warahmatullahi wabarakatuh. Sebelum kita tutup, mungkin bisa foto session dulu beberapa kali. Mungkin Mbak Iis bisa dipandu foto sessionnya.
Baik, terima kasih Pak Omar, Mas Omar sudah memandu acara hari ini. Mohon izin Bapak Ibu sebelum meninggalkan, yang tersisa minta tolong buka videonya kalau memungkinkan, kita foto bersama dulu ya sebagai dokumentasi. Baik, saya pandu. Oke, dari slide yang pertama, 1, 2, 3. Oke, slide yang kedua.
Cuman spasi doang.
1, 2, 3.
Tapi di sini apa ya? Daftar isinya ya.
1, 2, 3. Oke, wait. 1, 2, 3. Oke, next. 1, 2, 3. Oke. Dan baik, terima kasih sekali lagi Bapak dan Ibu atas kehadirannya. Kita bertemu lagi di next DSS seminar.
Terima kasih semuanya.
Terima kasih. Mohon izin.